We're going to briefly review the fundamental information regarding gain-of-function viruses and technology, CRISPR technology, genetic vaccines, and the requirements for us to take a completely different diagnostic and treatment approach in this new scientific era. This provides the foundation upon which scientists, physicians, and even patients can build upon using the information at the bottom of the slides, referencing each pieces of information you will see during this presentation. Everybody has gotten here by a different route. Whether you're a scientist trying to understand this, or a physician taking care of patients, or a patient who's had yourself an infection or disease or family members, it's important to understand that how we got here doesn't need to confuse us with where we can go as we improve our understanding and treatment of these disorders. While 1.1 million Americans are dead from COVID and 1.5 million have been injured due to the vaccines, and this number reflects greater numbers of deaths and injuries than the United States military has had since 1776. It's important at this point in time to get a better understanding and better approach to dealing with these genetic viruses. Many of us have come into conflict with individuals we care about, whether it be family members or friends or other members of the scientific community. It's time to put that behind us. It's time to grasp the information that we do have, and this presentation is designed for you to build upon so that we can move forward together. We're going to look at the viruses known as SARS-CoV-2, the inflammothrombotic disease known as COVID-19, and we're going to look at why people are having long-term consequences of post-acute sequelae, and the treatments based upon the best scientific medical evidence. We're going to look at a variety of topics, including how do we differentiate healthy from non-healthy tissue, how do we measure those differences, what are the origins of the viruses, what are treatments that we can confirm, how important is our immune system, what are the parts of our immune system, what are the 9 pathways that lead to cancer based upon these viruses and vaccines. And we're going to look at all of these in enough detail to make sense. We know that more than 500,000 excess deaths, in fact 540,000 excess deaths have occurred primarily due to inflammation and blood clotting, inflammothrombotic disease, heart disease, and cancers, high blood pressure, as well as pyogenic diseases, including Alzheimer's and other dementias. Few of them are the result of respiratory deaths. One of the concerns is the increasing rate of cancer diagnosis, and there's been a lot of questions debated as to the cause of this. One of the reasons is that people simply weren't being seen by doctors to screen, and we know that a later diagnosis leads to worse outcomes. This is an estimate of excess cancer deaths that was published by the Cancer Societies. You can see several publications here, and showing what looks like an almost plateauing effect of these cancers. So any increased excess deaths, we will have to look at seriously as a potential cause from the viruses and genetic vaccines. We know that the actual environment in which cancer cells find themselves are dependent upon those cancers having an increased metabolism, and that results in an increased blood flow to provide nutrients and to remove waste products through a variety of pathways. The process by which a cancer actually grows is not a linear process. There is the pathway in which the cells start to become increased metabolically active, and then they produce several chemicals, including vascular endothelial growth factor. This increases the blood supply, and you can see that as a result, there is a quadratic flow or an exponential increase as cancers get stronger and stronger, and this requires more than just a visual estimate of the disease. Here's a study that was published back in 2013 showing that radiologists simply looking at images can miss something so obvious as gorillas included on the chest CT, with only 24 of the radiologists actually noticing. Participating in the study, 83% failed to see the gorilla. We know that SARS-CoV-2 is associated with the recurrence of other viruses. So once the viral infection from SARS-CoV-2 attacks the immune system, other viruses that had been under control recur. We know that these viruses are associated with a number of health problems, including Epstein-Barr virus and herpes viruses, as you can see here, producing long-term sequelae. Those viruses cause further inflammation and blood clotting or inflammatory thrombosis through a variety of pathways, as you can see here, with several of these viruses having been identified over time, not only releasing certain chemicals, but being associated with certain cancers. So let's look at some of the foundational material that everybody should have a grasp on, particularly the scientific and medical community, and for the general public, extremely helpful for you to have this information when you talk with your doctor. The first step is actually trying to understand how we differentiate healthy and non-healthy tissue, and it is simply inflammation and blood clotting or inflammothrombotic changes that occur due to metabolism and regional blood flow changes. We can either look at this microscopically or we can look at it with nuclear imaging. This is the original theory that I presented explaining all of this at the American Heart Association in 1994. The graphic that you're looking at was put into a cardiology textbook in 1999. And as you can see, there's at least 16 factors that play different roles, including infectious diseases, vascular endothelial growth factor, and other growth factors. A simplified version of it looks like this and was also in the same textbook, where we explained that these inflammothrombotic diseases are heart disease, cancer, strokes, high blood pressure, diabetes, and infections, all a difference in metabolism and regional blood flow. I understand that for many people looking at this figure and the last one that you saw, that this is much like looking at Where's Waldo. There's an awful lot of trees in the forest, or a big forest with a lot of trees. And so part of the function of this presentation is to take and go look at each of the trees so that in the end it will be much easier for you to actually find where Waldo is. Although if you're having trouble, your second grader might be very helpful. We have been erroneously using blood tests over the course of many decades, primarily because it's what we've had. But by the time something shows up in blood, it's frequently a late-term consequence. And this research that we published back in 2008 shows that if we measure what's going on in the body with Fleming method compared to blood tests, we find out the blood tests are not very helpful. And many people have commented on the lack of helpfulness for symptoms and clinical findings in the case of the decision for what is COVID and what isn't. These symptoms are typical of any infection or cancer or a variety of other health problems, not very discreet and not very helpful in the end. The first information about vascular endothelial growth factor, as you can see here, was actually in the 1983 era, 1989 era. And this was the first evidence that we had that chemicals were being released that actually grew these new blood vessels. There's several types of vascular endothelial growth factor. In the end, inflammation and blood clotting cannot be separated, and it provides a balance against which we must treat patients, and we have to make decisions about how to treat them, nudging treatment versus pushing and breaking the system. As you look at this, the absence of disease is homeostasis, and either increased blood flow or decreased blood flow can result in a variety of different health problems, and we're going to look at these over the remainder of this presentation. This is again that diagram that you saw, the graphic from the book, and the right half of it was devoted to the effect of infectious diseases, bacteria, viruses, the like that cause. If you looked at this diagram in detail, you would see the innate immune system, the T cells, and the adaptive immune system, the B cells, producing chemicals and antibodies, and blood clotting effects producing inflammothrombotic response. This was a patient that was vaccinated and published in 2021 showing the inflammation. All the dark cells you see are inflammatory cells. The enlarged version that you see taken from the image is Congo red staining that looks for prion diseases in the heart, which is also a problem for these viruses and genetic vaccines. Fundamentally, The mitochondria that increase in more metabolically active cells like infection and cancer increase their activity and demand increased blood flow. But the mitochondria will take up the isotopes that we use in nuclear imaging, and the greater the blood flow on the right, as we see with cancers and infection, increase the delivery of the isotope, all of which we can use to differentiate tissue. We can either do it visually or we can quantify it for the first time with Fleming method, which is FMTVDM, which I will go through in a little detail here as we come through the slide series. The Fleming method for tissue and vascular differentiation and metabolism. Blumgart was the first person to actually use imaging qualitatively or visually. This was back in 1927. It wasn't until 2017 when I patented the method to quantify this, and the image to the far right with the heart in the 0.5 minutes was in the Federal Practitioner, the government's only peer-reviewed medical journal, where we showed that we could measure and distinguish tissue types. This is the patent for purposes of conflict of interest and clarification. If you look at the highlighted yellow, it says including the differentiation of tissue and metabolism. So this is Fleming method for tissue and vascular differentiation metabolism using same state singular sequential quantification comparisons. And it makes it possible to measure disease and treatment. This was early on. We showed that it was useful for heart disease and cancers. This is heart disease on the left and a breast cancer on the right, where we actually not only took the images, but then quantified or measured what was going on, which meant we could find cancer sooner, and we could then determine if your cancer treatment worked. The same was true for heart disease, and we were planning to launch this in 2019. FMTVDM, or Fleming method, allowed us to differentiate between normal, inflammation, infection, and cancer. And the thymus gland is highlighted. This was from a segment that we did on the Today Show and published in 2000, looking at bacterial infections of the heart. Treating the bacterial infections not only put the thymus back to rest, but reversed the heart disease. Both heart disease on the left and cancers and infection on the right follow a curvilinear pattern or a quadratic function pattern, where they are not just a straight line process. There is a change that occurs slowly and then more rapidly as the change continues, and we can measure that. It's important to look at this differentially from what we have been doing. Qualitative is like turning a light switch on, yes or no. This is where sensitivity and specificity errors occur, but quantitative measurements are accuracy. So once you've calibrated the system to be accurate, it's not a matter of whether it's right or not. It's a matter of where do you lie on the spectrum of health or disease. The origins of SARS-CoV-2. What we've been dealing with shows that there are three viruses, RS-3367, RS-SHC014, and MA15, that match genetically what we have been calling SARS-CoV-2. This is important because these three viruses are chimeric gain-of-function research viruses funded by several U.S. agencies, including the Department of Defense and Health and Human Services, with experts from Fort Detrick assisting with Peter Daszak at EcoHealth, along with the National Science Foundation and several other U.S. agencies. You can read about it if you like. And is COVID-19 a bioweapon? You decide. The evidence is there. More than $60 million was funded during that time. It's also important to note that the Department of Defense asked for a report from Ukrainian laboratories on a disease known as COVID 7 weeks before anybody was talking about viral infection SARS viruses in the Wuhan wet market. This is a copy of that report from U.S. Spending Government website. So it's very clear the U.S. was involved. You can see here's further evidence. They were in discussion. You can see this was November 11th and 12th of 2019. Again, 7 weeks before anybody was talking about a virus, the Department of Defense asked for this evidence, and there's no evidence of this website or documents being altered in any course since the 11th of November 2019. This simply shows that there was efforts taken to not report where this money was going to, and they were very interested in working on these processes, explaining perhaps why other countries have gotten so nervous about U.S. involvement in other countries and where our biological labs are. This is heavily redacted 2 emails that were sent to me back in 2021 before I released the book, Is COVID-19 a Bioweapon? This is where Fort Detrick was reaching out to me as a physicist, asking me to work with them on viral projects funded by NIAID. There's verification that, in fact, this Fort Detrick was looking for such individuals, imaging physicists like myself. This is the PCR data. You can see here several spots where the PCR matches for Wuhan HU1, the SARS-CoV-2 viruses, actually matches RS-3367, RS-SHC014, and CoV-MA15, all 3 viruses funded by these agencies through Peter Daszak, Ralph Barrick, and Shizang Li. Simply raising the question of remembering the oaths that you've taken and who you've taken them to, and our oaths have been to the general public. If you're an elected official, you took it to the general public in the country. If you're physicians, you took it to your patients and to those who trained you. But still, oaths are oaths that should be kept. This is the notorious spike protein, and what you're seeing here are the amino acids put together by different colors. There's 3 major parts of interest. One is the glycoprotein 120 segment. That's labeled number 1. Number 2 is the furin cleavage site. And number 3 is the area where the spike protein attaches to the ACE2 receptor, known as the regional binding receptor, and it has prion-like properties that we'll talk about. This was some of my initial work discussing the importance of how the different proteins and different structures like glycoprotein 120 attach to the sialic acid receptor. That's the first step that then allows the spike protein to be swung into contact with the ACE2 receptor. This was research I was doing looking at this inflammatory thrombotic disease. And you can see here further evidence and this is a cryo-electron microscopic evaluation. You can see why it's called a coronavirus, because if you slice through the virus, you see the little spike proteins coming out in what looks like a crown. This is the first part of the virus that attaches. The glycoprotein 120 attaches. It then swings the spike protein into contact with the ACE2 receptor. This is the 7th coronavirus known to infect human beings. Not the second, that's just simply the name that was given to it. We now want to take a look at the information we know about treating these viruses and confirming treatment response. Again, this required Fleming method to actually measure it, because you have a choice between either getting tissue or measuring it using Fleming method. We chose not to biopsy the heck out of everybody, but to measure the effect of treatments upon the virus and upon the inflammatory thrombotic disease known as COVID. This is a brief video talking about SARS-CoV-2.
This is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? And how would a vaccine work? The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, Droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes. Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then carries the spikes in vesicles which merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gather just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane. Now, with the virus spreading in your body, how can you develop pneumonia symptoms? For this, we'll have to look into your lungs. Each lung has separate sections called lobes. Normally, as you breathe, air moves freely through your trachea or windpipe, then through large tubes called bronchi, through smaller tubes called bronchioles, and finally into tiny sacs called alveoli. Your airways and alveoli are flexible and springy. When you breathe in, each air sac inflates like a small balloon. And when you exhale, the sacs deflate. Small blood vessels called capillaries surround your alveoli. Oxygen from the air you breathe passes into your capillaries, and then carbon dioxide from your body passes out of your capillaries into your alveoli so that your lungs can get rid of it when you exhale. Your airways catch most germs in the mucus that lines your trachea, bronchi, and bronchioles. In a healthy body, hair-like cilia lining the tubes constantly push the mucus and germs out of your airways, where you might expel them by coughing. Normally, cells of your immune system attack viruses and germs that make it past your mucus and cilia and enter your alveoli. However, if your immune system is weakened, like in the case of a coronavirus infection, the virus can overwhelm your immune cells, and your bronchioles and alveoli become inflamed as your immune system attacks the multiplying viruses. The inflammation can cause your alveoli to fill with fluid, making it difficult for your body to get the oxygen it needs. You could develop lobar pneumonia, where one lobe of your lungs is affected, or you could have bronchopneumonia that affects many areas of both lungs. Pneumonia may cause difficulty breathing, chest pain, coughing, fever and chills, confusion, headache, muscle pain, and fatigue. It can also lead to more serious complications.
Utilizing this approach to quantify tissue changes, research was conducted not only for the viruses SARS-CoV-2, but the inflammatory thrombotic response known as COVID-19. The details were laid out on the website NCT04349410 of the National Clinical Trials. And here you can see an image of coronavirus pneumonia, CVP, measured using Fleming method. You can see where this lies differentially to the right on the graphic, and you can also see tissue changes that occur with this type of inflammatory thrombotic response disease. Once hospitalized for patients who failed outpatient treatment, Fleming method was used to measure tissue changes. You can see on the female patient 3-1 who entered the study that azithromycin and hydroxychloroquine failed to improve her symptoms. Once hospitalized, she was placed on methylprednisolone or salumedrol. Follow-up 72 hours later showed improvement. Outpatient individuals were given an option of either receiving treatment or not. These were the 4 different drug treatment regimens that were given to individuals, in which their physicians reported their response, whether they thought they improved or not. You can see there were a variety of responses depending upon the particular regimen that was utilized. Individuals who failed and who were admitted to hospital were later analyzed to determine what did and did not work. It turned out if people had received antiminoquinolone prior to administration or hospitalization, either methylprednisolone or tocilizumab and interferon alpha 2 beta worked. If they hadn't received iminoquinoline, they received best response with the addition of those drugs, clindamycin, tocilizumab, and interferon alpha 2 beta. All the individuals in the study received immune support with the following vitamins and minerals, along with nebulizer treatments as necessary, and if necessary, heparin subcutaneous to address any potential thrombotic response. As you can see, vitamin C works on everything except the antigen presentation, but each of these vitamins and minerals have its unique benefit to the human immune system. All of the drugs that were selected for this research study were selected because they were known to have either a specific effect upon how the virus attaches itself, how the innate immune response and adaptive immune response occurred, and in fact what the oxygenation or adult respiratory distress syndrome, the fluid buildup in the lungs, responses were from different drugs. And you can see here wide variety of drugs were selected and then randomly assigned, as well as their mechanisms of action shown in the schematic or diagram on the far right side of this image. So independent upon what the drugs were called, they were selected for their mechanisms of action, which is, after all, what we're treating them with. You can see that specific drugs focusing on the attachment include clindamycin, hydroxychloroquine, primaquine, and zinc, all of which had different specific sites for viral attachment that they could affect. And measurement was made with Fleming method as to their outcomes, whether they did in fact respond. And then there was the additional question of how the spike protein would be cleaved and brought into the cells, and we can see that this is primarily the GP120, then the furin cleavage site splicing the S1, S2, and then the TMP or SS2 bringing the spike protein inside the cell for then viral replication and release. Inside the cell, infection basically was addressed by drugs that were known to affect the ribosomes, which is where the translation of the viral proteins occur and the reproduction of the virus, whether it be the mRNA, tRNA, or the ribosomes proper. This is further information about the protein translation. You can see how the different medications affect either tRNA or mRNA, so azithromycin, clindamycin, doxycycline, hydroxychloroquine, interferon alpha 2 beta, primaquine, remdesivir, and zinc. Each have a mechanism of action that was proposed, and that was the purpose for including them in the research study. Then there is the inflammothrombotic response, the initial T cell response, and the CD8 helper cells using major histocompatibility complex 1. This is not something I would expect the general public to know, but physicians and scientists should be aware of it. This part of the CD8 killer cell response was basically treated with 5 different drugs: clindamycin, hydroxychloroquine, interferon alpha 2 beta, methylprednisolone, and tocilizumab, each of which have a different response, as you can see here, depending upon different chemicals or toll receptors involved in this part of the innate T cell response, and then 5 drugs for the CD4 helper cells, the later responses, including clindamycin, hydroxychloroquine, interferon alpha 2 beta, methylprednisolone, and tocilizumab. So very similar drugs. You can see hydroxychloroquine also affects the thrombotic pathway by interfering with glycoprotein 2b3a and thrombus formation. Putting all the research together, the treatments that were shown scientifically to be effective were then published, as you noticed from that earlier slide, in multiple places, and they have been also reported in the Annals of Internal Medicine and other papers that have been published, quoting not only this research but the original inflammothrombotic response disease theory. We broke this down to individuals curious about the costs in immune support against SARS-CoV-2 pre-infection. Many people proposed these different vitamins and minerals. This turned out to be the cost of doing that for a month's supply, roughly $33 at the time in 2021. Once patients became symptomatic, the doses of those vitamins and minerals were increased, bringing it to just under $50 a month. Again, the data was noted earlier for the studies that have shown the effect of these vitamins and minerals on the human immune system. This looks at the breakdown of those 4 outpatient approaches and the costs. They're ranged anywhere from 40 some dollars to $95 per month at that point in time. Looking at several different combinations that were available, depending upon where you were at and what your physician was providing. For those who ended up hospitalized, you can see that the subsequent treatments with the steroids or interleukin 6 inhibitors or interferon inhibitors are shown here as you go through the sequence. Papers are out there available for research and also available on FlemingMethod.com. So instead of going through those in detail, I would simply refer you back to those. The cost of hospitalization actually in those treatments then ranged anywhere between $218 and $24,300, depending upon which regimen was selected at the time. So much less pricey to address it on the outpatient basis than it was on the inpatient, but still quite handleable throughout. It is important that individuals that are looking at the potential consequence of these post-acute sequelae and replication of the spike proteins, that it's important to note that step 2 focuses on the Wuhan HU1 spike protein, and mono treatment with these antibodies or even the vaccines that focus on Wuhan HU1 are not going to be as effective on current variants. The 5th step in this is looking at cancer. This was the original area of interest that people have had for the increased incidence, and we'll find that there are 9 pathways that are stimulated in this inflammothrombotic response disease known as cancer. Well, what is cancer? In some ways, I refer to it as teenagers or medical students. These are basically cells that are highly metabolically active. They require a lot of nutrients. They require a lot of blood supply, bringing those nutrients and moving the waste products, but they really don't provide much of a benefit for the human body. They are typically the result of damaged DNA, either due to viruses or some other factor, which we'll look at as we go through. The cell cycle is broken down into 2 cell growths known as G1 and G2, an area of interphase where the DNA is being synthesized, and then the division of the cells and mitosis. And on all of these steps, there are specific checkpoints where a variety of chemicals have been identified in the biochemical pathways, as you can see here, all of which are focused on making a decision about, is the process of this cell division going according to plan, or is the DNA damaged? And if it's damaged, how to address it, whether the cell should be aborted as such and killed, or whether it should be repaired. And that requires a biochemical process here. And I don't expect you to know all these, but it is important to point out that much of the changes that occur in the DNA are either due to a point mutation, some amplification of an area of DNA that shouldn't be, rearrangement of the chromosomes, or something happening to the cells, either through a methylation or acetylation process, which is a chemical reaction that occur from diet or UV radiation or viruses or drugs that change the DNA and alter it. Typically, the response, once altered, is to activate what's called an oncogene or a cancer-causing gene. These are named based upon where they were first recognized. So the rat sarcoma virus, or the myelocytomatosis gene, these are not just specifically for these cancers, but they are pro-cancerous genes when activated. The body also has inactivator pathways, so whether it's P53 or BRCA1 and 2, or the adenomatosis polyposis coli gene for colon cancer. In every one of these instances, these pathways are designed to stop the replication of the cell, to either repair it or kill it, so that the damaged DNA and the cancer does not continue to move forward. And this is broken down into several steps for the cell's survival, including motility, proliferation, viability, and differentiation circuits. Again, the changes in the genes are really the hallmarks and the body's response to recognizing when harm has been done to correct it. And you can see several of the pathways circled that we'll be talking about shortly. Again, this entire process is due to an increased metabolic function of the cell and increased blood flow as a result. It is this inflammothrombotic response that is being measured using Fleming method or FMTBDM, including tissue differentiation, metabolism. You can see vascular endothelial growth factor shows up prominently here, as all these other tissue factors encourage the growth. We know that a number of viruses are oncogenic. That means they promote cancer development. You can see several of them listed here in the types of cancers that they are associated with that have been documented over many decades of research. This paper was published in 2023, so a nice summary of just some of the major ones that we think of that we see being reactivated with the SARS-CoV-2 viruses. These viruses include both DNA viruses and RNA viruses. So you can see HIV is there present as certainly something there, and they upregulate or downregulate certain of those checkpoint factors and changes that promote aggressive growth of cells without function of the cells that we know as cancer. We know that you cannot vaccinate your way out of cancer. This is a human papillomavirus cancer, and as you look here, you can see the vaccination rates are up, the infection rates are down, but the behavior that's associated with the spread of these viruses and the subsequent development of these cancers continues to increase as the behavior increases, so you simply can't vaccinate away risky behavior. This is a nice example of measuring changes. This was a woman who actually took a soy product in an effort to reduce her risk of breast cancer. As you can see, these counts went from 108 to 130 to 184 to 240, showing that for this woman, she went from having normal breast tissue to ductal carcinoma in situ while taking a soy product. This clearly did not work for her, and this is part of the importance is measuring this. Well, how important is our immune system to our actual survival? Seems like that would be an obvious question, but let's take a look at some things that I think you may not be aware of. For example, women actually determine and select out partners that they're going to mate with or have children with based upon smell, and it's known that this smell is related to the immune system and the major histocompatibility genes. So it's thought that women can determine the smell of their partner to produce the best and most viable offspring just based upon smell and the immune system. And we know that this smell perception and behavior is affected by whether women are intending to have children, whether they're currently using contraceptives, and we know it also plays a role in fidelity issues as far as whether women will wander and look for other more sexually satisfying smell partners. We know that immunity is critical. This study was published in 2020 showing that individuals who actually had antibodies against the interferon that they made, and interferon interferes with viruses, individuals who had antibodies against their own interferon actually did worse and suffered worse COVID cases because they were unable to respond immunologically to the virus. We know that a number of the genetic diseases that occur with autoimmunity are associated with abnormalities in our immune system, whether it be T cells and B cells, as you can see on the left, and very specific markers of HLA, human leukocyte antigens, abnormalities, and diseases like celiac disease and others. Question is frequently being discussed about which type of immunity is better, natural versus vaccine. It's important to note that if you're given a vaccine, you're actually having or should be having a response again in your body that would imitate a natural response. The difference has to do primarily with what you're seeing in the vaccine versus what you're seeing with, quote, natural immunity. And here's an example of the antibodies and T cell responses made naturally. When we look at the natural person-to-person transmission, there are antibodies and immune responses to the spike protein, the envelope protein, the membrane, the nucleocapsid, which happens to be the one that humans have the greatest response to, but if you're only getting the spike protein, it's blunted. We've also been able to see that as the vaccines have frequently been given to people, something we haven't had for a disease before, there's a shift in the antibody response from the IgG3 that's protective to IgG4, and that increases with subsequent vaccines. This produces a response that's either considered an allergic response or an autoimmune response, which is completely different, and it results in a difference then of responding to something more like an allergen. And this allows the cancer cells to have an escape mechanism. So one of the problems here is that the repeated vaccinations are producing an IgG4 shift, and subsequent cancers are developing an escape mechanism, and they can evade IgG4. Is a nice example of evidence of that. These colored areas show different types of IgG levels, with IgG4 showing up in red. As you can see in the upper layer in the cancer areas, there are a lot of IgG4 showing up, where in the adjacent areas where it's non-cancerous or normal, there is no IgG4. So the presence of this IgG4 in areas of cancers has been shown to be associated with an increase in the growth, as you can see here. When IgG1 or IgG3 are present, the cancers aren't growing. When IgG4 is present, it allows the cancer to escape, and you can see this on the right-hand panels of both sides of these figures, where the addition of IgG4 allows these cancers to grow. So not a good thing as a result of the vaccinations. Homeostasis is talked about frequently with the balance, and immunity is no different. In the absence of disease, everything's in balance. On one end of the spectrum, you see cancers where there's decreased blood clotting and increased blood flow. To the left, increased clotting and decreased blood flow results in heart disease, diabetes, strokes, and even miscarriages, as we'll see later on in this presentation. Cancers can evade natural killer cells if they are allowed to produce chemicals to evade. And one of the concerns that's been expressed here with these continued vaccinations and suppression of the P53 system, which is important, have to do with CD74 and CD44. Again, I don't expect the general public to know this, but this is an interesting area for you to listen to. Here's a diagram showing research that was published earlier this year in 2023 looking at the various checkpoint pathways and interference that SARS-CoV-2 viruses do have that produce a change in the DNA of cells and DNA damage, producing inflammothrombosis and cellular senescence or injury. We know that there are many parts of the virus itself that plays a role in altering cells, including glucose metabolism and affecting the mitochondria, the powerhouses of cells, which are used uh, in nuclear imaging because of their uptake of these isotopes, allowing us to measure the effect of these viruses on cells. We've been talking about this for some point in time, or at least I have, as I've looked at this inflammothrombotic response. This was back from 2004 when we talked about the importance of looking at these inflammatory changes and actual changes leading to cancer. These changes can actually be measured using Fleming method as we quantify differences in metabolism and regional blood flow, allowing us to differentiate between inflammation, thrombosis, and cancer itself. Which brings us to the question of better understanding how these viruses and genetic vaccines, as well as any potential shedding of these spike proteins or other substances, as you'll see, lead to these long-term or post-acute sequelae consequences that we're now addressing, including cancer. To begin with, let's look at the various parts of our immune system. This will be a cursory review, certainly not in depth by any means, but enough to give a fundamental understanding. And it's important that we look at this from the homeostatic mechanism on putting everything in balance with this system. You can see that as blood flow actually increases and the clotting decreases, that we see cancers developing. On the other end of the spectrum, when we see an increase in blood clotting and decreased blood flow, we see things like myocardial infarctions, heart damage, strokes, diabetes, high blood pressure, and even miscarriages showing up. To understand that, I want to first take a look at the immune response to these viruses and what we must be looking at fundamentally with our immune system. I know that this is not one of those areas that most people enjoyed when they were in medical college or scientifically training, particularly medical college, and for the general public, I know it's not a very popular area, but it's important to just
breathe in through the nose, out the mouth. Wax on, wax off. Don't forget to breathe. Very important. Wax on, wax off. Wax on.
We're going to begin this actual presentation of the immune system by looking at the T cells, the innate T cells. And there's 5 major types: T helper 1, T helper 2, follicular helper cells, Th17 cells involved in autoimmune problems, as well as the T regulatory cells that say it's time to stop the actual immune response. We've got everything under control. Failure to do that also leads to autoimmune disorders. The first step here is then actually recognizing that there's a problem. The antigen presenting cells, which include natural killer cells, macrophages, and dendritic cells, find the material that's invading, in this case a virus, ingest it and tear it apart, and present the invader on their surface using major histocompatibility complex 2. This allows these cells to show the rest of the immune system there is a virus infecting you that you need to respond to. And you can see here there's a number of steps involved and chemicals, including CD28, interleukins, major histocompatibility complex 2, and a recognition of these T helper cells presenting the invader to the rest of the immune system in response for dealing with it. The second step is where these antigen presenting cells leave the site of infection, the lungs, the sinuses, and go to the lymph nodes. It's there in the lymph nodes that they release something known as interleukin 12 that takes naive T cells and turn them into T helper 1 cells. Here's a problem. You're naive, you haven't responded to anything, this is what you're going to respond to. And when this happens, there's a whole series of reactions that occur again through major histocompatibility complex 2 receptors to the naive T cells that turn them into T helper 1 cells using CD28. For those of you who've had the scientific and medical background, this is one of the things that we look for in cells to tell us what type of T helper 2 cell it is. Once activated, these T helper 1 cells release chemicals designed to kill and destroy anything infected, but it's very indiscriminate. It releases things like interferon, tissue necrosis factor alpha. It activates the cytotoxic CD8 helper cells, and it stimulates the production of T helper 2 cells. This is equivalent to just basically doing a bombing run and destroying whatever's underneath it. It's not discriminatory, it's not overly focused, and here you can see the T helper 1 cells releasing interleukin 12 as the process of trying to wipe out this viral invader in this case from the body, even at the cost of killing normal tissues, and which is where you become symptomatic with the runny nose, the fever, the headaches. The fourth step basically takes these cytokines 9 specific pathways that we know that the SARS-CoV-2 viruses and genetic vaccines are involved with as they yield cancer and other injuries to the body, including miscarriages and sudden cardiac death.